We are going to continue our discussion about protein folding. In the previous video, we talked about the primary and secondary structures involved in protein folding. In this video, I'm going to give you more detail about the tertiary and quaternary protein structure folding. So, there are two types of proteins. There are fibrous proteins and globular proteins. Fibrous protein are usually long sheets or strands, and one example is collagen. Globular proteins are usually spherical, and one example is myoglobin, and I would really encourage that you look up structures of these two proteins. Myoglobin is really important in your body in that it transports oxygen. It's kind of similar to hemoglobin, but it's only a single unit, whereas hemoglobin is 4 units, and myoglobin also has more affinity towards oxygen. Both of these protein folding structures are caused because of tertiary and quaternary protein folding interactions. So the first one that we're going to talk about is tertiary protein folding. The tertiary protein folding results in the 3D shape of the protein. Interactions that kind of hold the protein together include van der Waals forces, which are really weak interactions, and you can find these in almost any protein. Hydrophobic and hydrophilic interactions are basically when the hydrophobic or hydrogen or water-fearing elements of the amino acid side chains, they interact with each other, and they just stick together, and they don't like interacting with water, whereas the hydrophilic regions usually interact with water and with one another and kind of just are in the water exposed environment. And one important thing to note is that up to this point we've been thinking of interactions in the same side chain, like one or two amino acid residues, but in the main backbone of the amino group. But from here on, the tertiary and quaternary structure, you'll see that these interactions are usually between the R groups of amino acids. So these interactions are between the variable groups of the amino acid. Another interaction that is very common is electrostatic interaction, which is basically the anionic interacts with the cationic form of the amino acid. These are really common as well. And then one interaction that you can also see is disulfide bonds. So these disulfide bonds, what they do is they make the protein structure even more firm and tight, kind of like a linker, and they make them very strong. And this interaction is very specific to cysteine residues, and I will show you the structure of it in a second. So the cysteine residues, when they get oxidized to cysteine, and this forms the disulfide bridge that makes the interaction very, very strong. So here is the formation of the disulfide bond. The formation of the disulfide bond includes the loss of 2 protons, or hydrogen in this case, and the loss of 2 electrons. What I did is I wrote the hydrogens in black, and also the disulfide bond in black as well. So you'll see that these are 2 cysteine residues, which have a thiol group at the end with a hydrogen attached to it. And under the right conditions, you can lose those hydrogens and make a bond between the 2 cysteine residues to form. The residues are not going to be called cysteine. And then what this does is this gives the protein a more sturdy, more stronger structure, and it kind of prevents rotation. It kind of gives it that foundation to stay intact. And then in some cases, when you have a high pH, a high temperature, urea, or other denaturing conditions, you lose this structure, you lose the tertiary structure, and this process is called denaturation. The next structure is the quaternary structure, and this is the last structure in protein folding. Please make a note that not all proteins have this structure. In fact, there's not a lot of them that do, and it's not mandatory. The main defining characteristic of a quaternary structure are allosteric or cooperativity effect, and this is when you will see a sigmoidal curve, which I will show you in a second. The allosteric effects and cooperativity, what it basically says is that the binding of a molecule to one of the subunits of the quaternary structure will determine the level of molecules affinity to the other subunits. So for example, if I have 4 subunits and I have, let's talk about hemoglobin. Hemoglobin has 4 subunits, so we would say that it has a quaternary structure because it has more than one subunit. There's more than one subunits interacting with each other. So the binding of oxygen to one unit, say subunit 1, increases the affinity for oxygen in subunit 2, and then when another oxygen binds to subunit 2, the third subunit will have even more higher affinity towards the oxygen. So this is kind of like a cycle, and it also works the other way around. It doesn't necessarily mean that every single time you have a subunit, one of them is going to increase the affinity for the other subunit. No, you can have something that binds to subunit 1 and decreases the affinity for the other subunits, and it can work that way as well. Like I said, not all amino acids have this structure, and in this interactions, in this structure, you have the same interactions as is the tertiary structure. So you still have hydrophobic and hydrophilic interactions, you have electrostatic interactions, you have van der Waals interactions, and you might even have disulfide bond interactions going on in here. The next topic is called conjugated proteins. So what conjugated proteins are is that they derive their function from a covalently attached molecule. So and these groups are usually called prosthetic groups, and what these prosthetic groups are just basically covalently or permanently or temporarily attached groups that increase or decrease the function of the protein. So you can have organic prosthetic groups, and when I mean organic, I mean vitamins and metal ions. One common example is hemoglobin. The prosthetic group it has is iron, and it really requires that for its function. Similarly, if you have a lipid attachment to a protein, they're called lipoproteins. If you have a carbohydrate attachment, they're called glycoproteins, and if you have a nucleic acid attachment, they're called nucleoproteins. These groups, they enhance the function of the protein or the structure that you're talking about. They don't necessarily mean that they have to have it, and that's all the time. Some proteins, they just do better in the presence of these modifications, and if the modifications are absent, they still do their role, but not as efficiently. One last topic from this unit is a concept called denaturation, and what this is basically the loss of any of the structures of protein folding. These include the secondary, tertiary, and quaternary, but the primary structure still stays intact, and denaturation usually happens when you have an increase in salt concentration, pH, or an increase in temperature or decrease in temperature, or you add a denaturing agent like urea. This can lead to loss of function of the protein. When I mean proteins, most proteins are enzymes, but not all proteins are enzymes, so it can lead to some kind of loss in function if it happens in the right area and under the right circumstances.